Citrate cycle. The citrate cycle is the metabolic engine of the cell. When energy in the cell is low, the citrate cycle is critical to the production of energy from fatty acids, proteins, and carbohydrates. The fuel for the citrate cycle is acetyl-CoA. The exhaust is carbon dioxide, and the work performed is the transfer of electrons. In each turn of the citrate cycle, one acetyl-CoA is oxidized, and 8 electrons are transferred to 3 NADH and 1 FADH2. In the process, 1 GTP is also produced by direct phosphorylation of GDP. The NADH and FADH2 are used to power the electron transport system, resulting in the production of approximately 10 ATP for each acetyl-CoA that enters the citrate cycle. The flow of acetyl-CoA into the citrate cycle is controlled by the pyruvate dehydrogenase enzyme complex. The pyruvate dehydrogenase complex has a molecular mass of almost 8,000 kilodaltons and contains nearly 80 individual protein subunits. Pyruvate dehydrogenase uses pyruvate from the glycolysis reactions and metabolizes it to acetyl-CoA, fueling the citrate cycle. The pyruvate dehydrogenase complex consists of 3 interdependent enzyme subunits that use pyruvate, CoA, and NAD+ as substrates, and in a 5-step reaction mechanism, generates carbon dioxide, acetyl-CoA, and NADH. 5 different coenzymes are needed to enable the enzymatic activity of the pyruvate dehydrogenase complex. Nicotinamide adenine dinucleotide, derived from vitamin B3, flavin adenine dinucleotide, derived from vitamin B2, coenzyme A, derived from vitamin B5, thiamine pyrophosphate, derived from vitamin B1, and lipoamide. The product of the pyruvate dehydrogenase reaction, acetyl-CoA, is used to fuel the citrate cycle. The first reaction in the citrate cycle commits to the acetate unit of acetyl-CoA to oxidative decarboxylation by catalyzing its addition to oxaloacetate, forming citrate. Oxaloacetate is regenerated in the last step of the cycle, so it can be reused as the substrate for the first step. Note that the metabolic cycle is referred to here as the citrate cycle because citrate exists predominantly in the deprotonated form at physiological pH and is therefore not an acid in living cells. Other names for the citrate cycle are the tricarboxylic acid cycle, or simply TCA cycle, and the Krebs cycle, named after the biochemist who first described it, Hans Krebs. The citrate cycle contains 4 coupled redox reactions. They use the oxidized forms of the coenzymes NAD+ and FAD as electron carriers, reducing them to NADH and FADH2. NADH and FADH2 are then oxidized by the electron transport system, producing ATP. Citric acid is used commercially for a wide variety of purposes. It can be isolated in great quantities from the fungal mold Aspergillus niger using fermentation methods that exploit the citrate cycle reactions. High levels of citrate synthase are found naturally in the fungus. When the other citrate cycle reactions are inhibited under certain culture conditions, citrate is overproduced by the fungus and secreted into the culture medium.